Hello students, myself Dr. Prasoon Hara. We will study today porphyrins. One of the most important ways in which metal ions are involved in complexes with a type of macrocyclic ligands called porphyrins. Porphyrins are derivatives of porphyrins. They differ in arrangement of substituent around the periphery. The porphyrin molecule consists of macrocyclic pyrrole system with conjugated double bond and various groups attached to perimeter. Porphyrin group can lose two protons to form the two diion which form the porphyrin complex with metal ion which are known as metalloporphyrins. Few most important metalloporphyrins include heme and chlorophyll. Heme is the important constituent of hemoglobin and myoglobin, the important oxygen carrier proteins. Now, let us discuss structural and the working of myoglobin and hemoglobin. Hemoglobin and myoglobin. Hemoglobin, which is represented by Hb, and myoglobin represented by Mb. They are responsible for the transport and storage of oxygen in higher animals. Like mammals, hemoglobin transports oxygen from its source to the site of its use inside the muscle cells, where oxygen is transferred to myoglobin for use in mitochondrial oxidation. Myoglobin is a monomeric protein with molecular weight 17,100, having a single polypeptide chain that is not conducive of self-association. On the other hand, hemoglobin is a tetrameric protein. Hemoglobin is a tetrameric protein and with molecular weight 64,500, consisting of two alpha and two beta peptide chains interlinked through hydrogen bonded COO minus NH interactions. Due to these salt bridge interaction, the peptide chain in deoxyhemoglobin is constrained. X-ray diffraction study showed the disappearance of these salt bridges on oxygenation. The active site of both hemoglobin and myoglobin contain the heme group, in which Fe2 is equatorially coordinated by the four pyrrole nitrogen atoms of porphyrins. Like we can see in this diagram 1, 2, 3 and these four pyrrole rings. The fifth position is coordinated by the imidazole nitrogen atom of histidine of the protein chain. The sixth position is vacant but hydrophobically shielded by the protein chain. As a result, only non-polar neutral molecules such as oxygen, carbon dioxide, etc. can bind to sixth position, due to which iron is approximately 0.75 angstrom above the level of the porphyrin ring. This is as shown in this diagram. It is in the center there is an iron atom above the level of porphyrin ring. Thus, in the deoxygenated hemoglobin, Fe2 plus form high spin complex with 4 unpaired electrons and square pyramidal geometry. In this structure, we can see the structure of deoxygenated hemoglobin unit. Deoxygenated hemoglobin unit. One of the four subunits of hemoglobin, position of heme group between the two hexacle of hydrophobic globin protein. The two hexacle of hydrophobic globin protein. And subunit of hemoglobin are here. This is the 0.75 angstrom. Here is the hydrogen and here protein molecule is there. As square pyramidal geometry, we can see it very clearly. The structure of globin in fact consists of 8 hexagonal parts. Heme group is present between two of the hexagonal part as shown in the above this two hexagonal part, this one and this one. Globin protein consists of two parts, polar and non-polar. Polar end of the protein is projected outside and non-polar inside around the heme group. In the absence of globin, the sixth position is coordinated by polar molecule like water molecule. Here, like water molecule and Fe2. Heme is irreversible, oxidized by oxygen of air to Fe3, hematine. The latter, because of its residual positive charge, is reluctant to bind uncharged ligands such as oxygen, but readily binds to charged ligands such as Cn-, S2-, OH-, etc., which inhibit oxygenation. Oxygen bind to Fe2 heme at the vacant sixth position, and resulting octahedral field is strong enough to transform high spin Fe2 to low spin Fe2, where the radius is 0.75 angstrom. As a result of that, Fe2 radius is contracted by about 0.17 and Fe2 in the active site heme of oxyhemoglobin, and oxymyoglobin moves towards the porphyrin plane and ultimately sits in the porphyrin cavity. Uh, we can see in this diagram the structure of subunit of oxyhemoglobin, in which the iron is in the plane of heme. Yes, it's the same plane in the for the heme molecule. The movement of Fe2 causes the coordinated histidine to move towards the porphyrin plane. This brings about a conformational change throughout the peptide chain, amounting to rupture of some of all the COO minus NH3 salt bridge interactions. The constrained hemoglobin tetramer then relaxes by exposing the sixth position of the remaining three units of the heme groups to oxygenation. This phenomenon is known as interaction. This phenomenon is known as cooperative interaction. Oxygenation of hemoglobin is autocatalytic due to these cooperative interactions. But such effect is absent in myoglobin due to its monomeric structure. The overall rate of binding of ligands to hemoglobin is much slower than that of myoglobin, an isolated hemoglobin subunit. In vertebrates, dioxygen enters the blood in the lungs or gills, where the partial pressure of dioxygen is relatively high, approximately 160 millimeters Hg. It is then carried to the tissues under ideal conditions, where the partial pressure value is 20 to 50 mmHg, which is considerably lower than lungs. The reactions are as follows. Hemoglobin plus oxygen gives oxyhemoglobin, and oxyhemoglobin plus myoglobin gives oxymyoglobin plus hemoglobin. Hemoglobin has an ambivalent functions. It should bind dioxygen tightly and carry as much as possible to the tissues, but once there it should relinquish it readily to myoglobin, which can store it for oxidation of foodstuffs. Myoglobin must have greater affinity for dioxygen than hemoglobin in order to effect the transfer of oxygen from oxyhemoglobin to myoglobin. In cell, oxygen affinity of myoglobin must be higher than that of hemoglobin. Due to monomeric nature and the absence of cooperative interaction, myoglobin take up oxygen in 1:1 molar ratio. Here myoglobin plus oxygen gives oxymyoglobin. Kmb is equal to MbO2 whole twice divided by MbPO2. If F represents the fraction of myoglobin, molecule bearing oxygen and P represents the equilibrium partial pressure of oxygen. Then K is equal to F F upon 1 minus Fp, and F is equal to Kp upon 1 plus Kp. This is the equation for hyperbolic curve, which is shown in the lower figure. We will see this. Hemoglobin with its 4 subunits has more complex behavior. It approximately follows the equation F is equal to Kp to the power n, 1 plus Kp to the power n is equal to 2.8, where the exact value of n depends on pH. Thus, for hemoglobin, the oxygen binding curves are sigmoidal. The fact that n exceeds unity can be ascribed physically to the fact that attachment of oxygen to one heme group increases the binding constant for the next O2, which in turn increases the constant for next one, and so on. Although hemoglobin is about as good as oxygen binder as myoglobin at high oxygen pressure. It is much poorer at lower pressure, prevailing in muscles, and hence passes on its oxygen to myoglobin as required. Moreover, the need for oxygen will be greatest in tissues that have already consumed oxygen and simultaneously have produced carbon dioxide. The CO2 lower the pH, thus causing the hemoglobin to release even more oxygen to the myoglobin. The pH sensitivity, which is called the Bohr effect, as well as the progressive increase of the oxygen binding constants in hemoglobin, is due to the interactions between the subunits. It is clear that each of the two is essential in the complete oxygen transport process. We can see here the oxygen binding curve for myoglobin and hemoglobin, showing also the pH dependence, which is a Bohr effect for the latter. Is the percentage saturation is showing on the y-axis, and partial pressure showing in muscles, and that is in lungs. It's a myoglobin curve. This is hemoglobin curve at pH is equal to 7.6 and hemoglobin curve pH at 6.8. Carbon monoxide, PF3 and few other substances are toxic because they becomes bound to the iron atom of the hemoglobin more strongly than oxygen. Their effect is one of the competitive inhibition. That's all. Thank you.